Right now, every single human being alive today shares a truly remarkable genetic connection, tracing back through an unbroken maternal lineage to one specific ancient woman researchers have named Mitochondrial Eve. This particular scientific finding sounds almost mythological, but it actually represents a well-documented, carefully established genetic conclusion worth exploring properly, along with several important clarifications regarding exactly what this finding actually means, and just as importantly, what it doesn't mean. Let's dig into this properly because this represents one of the more frequently misunderstood findings in modern genetics, and untangling the actual science reveals something seriously fascinating about how researchers study deep human ancestry. Let's start with the crucial clarification worth establishing clearly up front, because it directly corrects a common, persistent misconception surrounding this specific topic. Mitochondrial Eve wasn't the only woman alive during her specific historical period, and she wasn't some kind of biblical first human woman in any literal sense. She represents the most recent common ancestor from whom every currently living human inherits their mitochondrial DNA, specifically a very particular, narrow category of genetic inheritance passed down exclusively through the maternal line, generation after generation, entirely separate from the vast majority of a person's genetic material inherited through both parents combined. This detail matters enormously for properly understanding this entire topic. Mitochondrial Eve lived among a considerable population of other humans during her own specific historical period, with many other women from that same population also having children and passing along their own genetic material. The key distinction is that Mitochondrial Eve happens to be the specific individual whose particular mitochondrial DNA lineage, traced purely through an unbroken chain of mothers to daughters to granddaughters and so on, happens to be the only maternal lineage that has survived uninterrupted all the way through to every single human currently alive today. Let's talk about mitochondrial DNA specifically, because understanding this particular genetic material's unique inheritance pattern is essential for properly grasping why this specific finding works the way it does. Mitochondrial DNA, found within mitochondria, the small cellular structures responsible for generating cellular energy, gets passed down exclusively through the maternal line, since a developing embryo's mitochondria come almost entirely from the egg cell specifically, with the sperm cell's own mitochondria generally not surviving the fertilization process in any meaningful, ongoing way. This means mitochondrial DNA provides researchers with a remarkably clean, uncomplicated genetic marker for tracing purely maternal ancestry, unlike most other genetic material, which gets shuffled and combined from both parents during ordinary sexual reproduction. Let's talk about why some maternal lineages eventually disappear over successive generations too, because this represents an important underlying concept worth exploring in additional depth. If a woman has only sons and no daughters, her specific mitochondrial DNA lineage ends with that particular generation, since sons don't pass mitochondrial DNA along to their own children. Over enormously long timescales, spanning countless successive generations, the overwhelming majority of maternal lineages from any given ancient population will eventually die out entirely through this same basic process, simply due to the accumulated statistical likelihood of any given specific lineage eventually producing only sons, or no surviving children at all, at some particular point across this extended time frame. This detail helps explain the underlying mathematical and genetic logic behind mitochondrial Eve's existence. Given enough successive generations, purely through this ongoing process of lineage extinction already discussed, the number of surviving distinct maternal lineages from any sufficiently ancient population will naturally, gradually narrow down, until eventually only a single maternal lineage remains that connects, without any interruption, to every currently living descendant of that original ancient population. Let's talk about when mitochondrial Eve actually lived, because this represents an important, carefully researched detail worth exploring specifically. Genetic research indicates mitochondrial Eve lived in Africa roughly 150 to 200,000 years ago, placing her within the same general early period already explored extensively in other discussions regarding early modern human evolutionary history and origins. This timeline is worth appreciating because it directly reinforces the broader scientific consensus already established through completely separate lines of fossil and archaeological evidence, confirming Africa as humanity's evolutionary birthplace, with this specific genetic finding providing an entirely independent line of scientific evidence pointing toward the exact same fundamental conclusion. Let's talk about how researchers actually calculated and confirmed this specific finding too, because this represents an important methodological consideration worth addressing directly. Researchers analyzed mitochondrial DNA samples collected from populations across the entire world, carefully comparing the specific genetic differences and mutations accumulated within different population's mitochondrial DNA over successive generations. Since mitochondrial DNA mutations accumulate at a relatively steady, well-documented rate over time, researchers could use these accumulated genetic differences to construct a detailed genetic family tree, eventually tracing every single sampled mitochondrial lineage back to one single common ancestral point, allowing researchers to both confirm this shared common ancestry and estimate roughly when this specific common ancestor likely lived. Let's talk about the equivalent paternal lineage concept too, because this represents an interesting, closely related finding worth exploring, specifically extending this same basic genetic logic to paternal ancestry as well. Researchers have similarly identified what's sometimes called Y-chromosomal Adam, representing the most recent common ancestor from whom every currently living man inherits their Y chromosome, tracing an equivalent, unbroken paternal lineage in broadly the same way already discussed extensively throughout this topic regarding maternal lineage and mitochondrial DNA specifically. Interestingly, current genetic research suggests this particular paternal common ancestor likely lived at a somewhat different specific time compared to Mitochondrial Eve, illustrating that these two separate genetic tracing methods, while conceptually similar, don't necessarily point toward the exact same specific individual or even precisely the same historical time period. Let's talk about why this detail regarding different timing matters so significantly too, because it reinforces the crucial clarification already established at the very beginning of this discussion. The fact that Mitochondrial Eve and Y-chromosomal Adam likely lived at somewhat different specific times directly confirms that these two individuals almost certainly weren't a literal couple living together, further reinforcing that these specific genetic findings represent statistical, genealogical conclusions regarding surviving lineages specifically, rather than identifying some kind of literal single original human couple from which all subsequent humanity directly descended in any simple, straightforward sense. Let's talk about mitochondrial DNA's broader practical applications beyond simply this specific ancestral finding too, because this represents an interesting, practically relevant dimension worth exploring before continuing this broader discussion. Beyond tracing deep evolutionary ancestry already discussed extensively throughout this topic, mitochondrial DNA analysis has proven remarkably valuable across numerous other practical applications, including forensic investigation, since mitochondrial DNA survives considerably better than other genetic material in degraded biological samples, and genealogical research, allowing individuals to trace their own specific maternal ancestry considerably further back than conventional genealogical records typically allow, sometimes revealing detailed information regarding specific ancient population movements and migrations connected to that individual's own particular maternal lineage. Let's talk about haplogroups specifically too, because this represents an important, closely related genetic concept worth exploring in additional depth. Researchers studying mitochondrial DNA have identified and mapped numerous distinct haplogroups, essentially specific genetic lineage branches that developed as different populations gradually spread across different world regions following humanity's initial migration out of Africa. Each haplogroup carrying its own specific, identifiable pattern of accumulated genetic mutations that researchers can trace back to specific approximate geographic origins and historical time periods. This detailed haplogroup mapping has allowed researchers to construct an increasingly precise, detailed picture of humanity's broader migratory history following our species' initial African origin, directly connecting individual genetic ancestry testing results to this same broader scientific framework already established through the Mitochondrial Eve research discussed extensively throughout the rest of this topic. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Let's talk about how this specific mitochondrial DNA research connects directly to the broader out-of-Africa migration theory too, because this represents an important, closely related connection worth exploring specifically. The detailed haplogroup mapping already discussed provides strong, independent genetic support for the broader scientific consensus already established through fossil and archaeological evidence, confirming that modern humans originated in Africa before eventually migrating outward to populate the rest of the world, with mitochondrial DNA haplogroup diversity actually being considerably higher within within African populations, specifically compared to populations elsewhere in the world, a pattern entirely consistent with Africa representing the original source population from which smaller migrating groups eventually departed, carrying only a subset of the original population's total genetic diversity along with them. Let's talk about the specific mutation rate researchers rely upon to calculate these ancestral timelines too, because this represents an important, technically detailed methodological consideration worth exploring before continuing this broader discussion. Researchers have carefully calibrated the specific rate at which mitochondrial DNA mutations tend to accumulate over successive generations, using a combination of known historical population divergence events and direct observation of mutation rates within modern populations with well-documented genealogical records, allowing researchers to convert the specific number of accumulated genetic differences between different populations into a reasonably confident estimate regarding roughly how many generations and correspondingly how many years have actually passed since those populations shared their most recent common maternal ancestor. Let's talk about scientific debate and ongoing refinement regarding this specific research area too, because it's worth acknowledging honestly that this particular scientific finding, like most active areas of ongoing genetic research, continues receiving refinement and, in certain specific respects, adjustment, as researchers develop increasingly sophisticated analytical techniques and gather additional genetic samples from previously underrepresented populations. Earlier estimates regarding mitochondrial Eve's specific approximate living period have been refined somewhat over time, as genetic sequencing technology and analytical methodology have continued improving, representing a normal, healthy feature of ongoing active scientific research rather than any fundamental uncertainty regarding the core underlying concept and broader conclusion already established and confirmed through multiple independent lines of supporting genetic evidence. Let's talk about popular media and cultural reception of this specific scientific finding too, because this represents an interesting, closely related dimension worth exploring specifically, directly connecting to the persistent misconception already addressed at the very beginning of this discussion. When researchers first announced this particular finding, popular media coverage frequently emphasized the catchy, biblically evocative Eve naming convention without adequately explaining the crucial scientific nuance already discussed extensively throughout this topic, contributing directly to the persistent popular misconception treating mitochondrial Eve as some kind of singular original human ancestor, rather than accurately representing her as simply the specific individual whose particular maternal genetic lineage happened to survive uninterrupted through countless subsequent generations of ongoing human reproduction and lineage extinction. Let's talk about how this specific finding will actually continue to change over future time too, because this represents a really fascinating, often overlooked dimension worth exploring before continuing this broader discussion. Mitochondrial Eve's specific identity isn't permanently fixed for all future time, as additional maternal lineages continue naturally dying out over future generations through the same basic process already discussed extensively throughout this topic. The most recent common maternal ancestor connecting all future humans will gradually shift forward in time, meaning some future population of humans, many generations from now, will trace their own common maternal ancestor back to a woman who, from our own current perspective today, would actually be one of mitochondrial Eve's own descendants rather than mitochondrial Eve herself. Let's talk about how ancient DNA research from actual skeletal remains has provided additional, independent confirmation of this broader picture too, because this represents an important, more recently developed complementary research approach worth exploring in additional depth. Beyond studying mitochondrial DNA within currently living populations already discussed extensively throughout this topic, researchers have increasingly succeeded in extracting and analyzing genetic material directly from ancient human skeletal remains, allowing for direct genetic comparison between actual ancient populations and modern populations, rather than relying purely on inference drawn backward from modern genetic diversity alone. This ancient DNA research has generally provided strong additional confirmation for the broader migratory and ancestral picture already established through modern population genetic research, while also occasionally revealing additional nuance and complexity, including evidence of ancient population movements and genetic mixing events that purely modern population-based genetic research alone couldn't have fully, directly confirmed without this kind of direct ancient genetic sampling and comparison. Let's talk about common related misconceptions regarding human genetic diversity more broadly too, because this represents an important, closely connected dimension worth addressing directly before continuing this broader discussion. The mitochondrial Eve finding sometimes gets misinterpreted as suggesting humans are somehow less genetically diverse than commonly assumed, when the actual scientific reality is precisely the opposite in certain important respects. While all humans do share this remarkably recent common maternal ancestor already discussed extensively throughout this topic, human genetic diversity more broadly, considering the vast majority of a person's total genetic material beyond simply mitochondrial DNA specifically, remains considerable, reflecting many thousands of generations of subsequent genetic mixing, mutation, and regional adaptation occurring since mitochondrial Eve's own specific lifetime. This detail is worth including because it helps prevent an easy, though inaccurate, oversimplification sometimes drawn from this particular finding, clarifying that shared recent maternal ancestry regarding this one very specific narrow category of genetic inheritance doesn't actually imply limited overall human genetic diversity more broadly considered. Let's talk about how this research connects to broader philosophical and cultural questions regarding human unity and shared ancestry too, because this represents a truly interesting, more reflective dimension worth exploring before this discussion concludes. Beyond its purely scientific significance already explored extensively throughout this topic, the mitochondrial Eve finding has attracted considerable broader cultural and philosophical interest specifically because it provides a scientifically grounded, genetically confirmed basis for emphasizing humanity's shared common ancestry and fundamental biological unity, offering a compelling scientific counterpoint to any assumption that different human populations represent fundamentally deeply separate biological lineages rather than representing relatively recent branches from this same shared, remarkably recent common ancestral root already thoroughly documented and confirmed throughout this discussion. Let's talk about how researchers continue expanding and refining this specific research area through increasingly diverse global sampling too, because this represents an important, actively ongoing research priority worth exploring specifically. Earlier mitochondrial DNA research sometimes relied on comparatively limited, geographically unrepresentative population sampling, and researchers have subsequently worked to considerably expand genetic sampling across a much broader, more seriously representative range of global populations, helping ensure this research area's specific conclusions rest on increasingly comprehensive, increasingly reliable underlying genetic data rather than potentially skewed or incomplete earlier sampling that might have inadvertently produced less accurate or less representative conclusions regarding humanity's actual broader genetic history and diversity. Let's talk about the specific technical challenges involved in reliably dating events this far back in human history too, because it's worth acknowledging honestly before wrapping up this discussion that estimating precise dates for events occurring many tens of thousands of years in the past inevitably involves meaningful statistical uncertainty and a reasonably wide potential margin of error. Researchers generally present these kinds of ancient genetic timeline estimates as reasonably confident ranges rather than single, precisely fixed dates, reflecting appropriate scientific humility regarding the genuine methodological challenges involved in reliably reconstructing detailed genetic history, extending this far back into humanity's deep evolutionary past, using indirect genetic evidence rather than any kind of direct historical documentation or observation. Let's talk about how this whole finding connects to broader questions regarding human genetic diversity and shared ancestry too, because this represents an important, unifying point worth exploring before wrapping up this broader discussion. Despite humanity's considerable visible physical diversity across different populations worldwide, this specific genetic finding reinforces a broader scientific conclusion already well established through numerous other independent lines of genetic evidence. All currently living humans share remarkably recent common ancestry, considerably more recent and considerably more thoroughly interconnected than the visible physical diversity across different human populations might otherwise casually suggest. Let's talk briefly about how ordinary people can actually access and explore their own connection to this broader scientific finding today too, because this represents a fitting, practically accessible final point worth including before this discussion concludes. Various commercial genetic testing services now offer individuals the opportunity to have their own mitochondrial DNA analyzed, allowing interested people to identify their own specific maternal haplogroup and trace their own personal maternal lineage back through this same broader scientific framework already explored extensively throughout this discussion. This detail is worth appreciating because it demonstrates how what once represented highly specialized, cutting cutting-edge academic genetic research has gradually become considerably more accessible to ordinary interested individuals, allowing people today to directly, personally engage with and explore their own specific place within this remarkable, scientifically documented shared human genetic history. Let's zoom out and pull all of these different threads together, because the full picture of who Mitochondrial Eve actually was turns out to represent a carefully documented, truly fascinating genetic finding, though one requiring careful clarification to properly avoid common, persistent misunderstanding. She represents the most recent common ancestor connecting every currently living human through an unbroken maternal genetic lineage. Living in Africa roughly 150 to 200,000 years ago, among a considerable broader population of other contemporaries, rather than representing some kind of singular, isolated original human ancestor in any simple, literal sense. So the next time this truly fascinating scientific finding comes up in conversation, it's worth sharing the accurate, complete picture, a specific woman whose particular maternal genetic lineage happened to survive uninterrupted through countless subsequent generations, rather than the common, oversimplified misconception treating her as some kind of singular, biblical first human ancestor, which leaves an interesting thought worth sitting with. If something as seemingly straightforward as tracing human ancestry back to a single common ancestor actually requires this much careful scientific nuance and clarification to properly understand, what does that suggest about how frequently seriously fascinating scientific findings become oversimplified or distorted as they spread through popular culture and casual conversation, gradually losing the careful nuance that made the original findings scientifically accurate and meaningful in the first place?